kind of work pretty well with the surveys on either side.
Yes, I'm knocking on a fake.
Yeah, I guess that's it. It should be fine.
Yeah.
During the lecture, we can probably just.
No, definitely don't want that. Yes, thank you. So,
Can you chill, okay? Just relax, Jason. It's gonna be fun.
Oh, Professor Chu, these are all UC Dragon Boat people. And Jeffrey, too.
Yeah, we're all on the same team.
You just no comments from Professor Chu?
Yeah. Where you guys sit?
okay, folks, we're going to get going in a few minutes. So if you can get out some kind, turn on your notes, get your notes out, whatever works best for you. If you do have a Wi-Fi enabled device, if you can log on at some point. We're not going to use Notonic right away because not everyone has downloaded it. In fact, if you do have questions about Notonic, the Notonic team is here at the front table. They will also be available during the break, so you can ask them. So you don't have to worry about participation marks involving Notonic right now. So you can just get on to menti.com and type in that code. Hey, Sarah.
Your business card thing?
Oh, I did not bring. Maybe I have one. I don't know.
I hope so.
I might. I don't know. I do.
Thank you so much.
No problem. Okay, so one of you guys has to be fairly close by in case something does not go. Okay, sure. Okay, that makes sense. Awesome. All right.
James is staying too, so he'll be just if
Okay.
you need him.
Yep. Okay. Thanks, James.
Bang on the door, I'll be right here.
All right. Okay, so thank you. Before we get going, we do have a couple of announcements for you. I had posted it earlier on the announcements page, but I just wanted to draw your attention to a couple of things that are coming up. So the first thing is, if you do have things that you can spare and share in terms of non-perishable food items, the food bank is kind of running right now, and as the week goes on, it's going to start needing some replenishment. A lot of your colleagues are kind of suffering from the same OSAP cuts that you are. So if you do happen to have something that you can donate, there is a donation box in the HMB office. Don't feel obligated, but if you do have the opportunity, please go ahead and do so. And then I'm going to turn this over to Marlene and Delphine, and they'll tell you a little bit about what other events you might be interested in.
Nice, thank you. Hi everyone, I'm Delphine, one of your co-presidents this year for the Human Biology Students Union.
And my name is Marlene, I'm the other co-president.
So we're just going to tell you about some events that are coming up. Happening tomorrow outside the UTSU front lawn, we'll be hosting a free barbecue. So all you have to do is show your acorn, prove that you're in lifestyle or HMB, and then you'll be good to go with a free burger.
Yeah, working
go. Thank you.
Thanks for pressing G.
All right, thanks for the announcements. So, it's not streaming at all? Okay, so while we're waiting for some of the technical glitches, yep, we're good. Okay, awesome. Thank you for doing that. And if you guys don't mind checking up on the stream, if there are any questions that are coming from the online group, please let me know. So what we're kind of going through today is going back and revisiting this emerging hypothesis. And I've posted some papers over the last little while. They're for your own interest. If you're interested in this field and trying to get established in this field, these papers that are coming out are actually more or less in line with what I'm going to be telling you today. So we're going to go back, talk about what happens to your normal gut microbiome that you have developed, and you have changed your gut microbiome. If all of the science is correct, you have had 3 different times in your life where your microbiome has changed. And the one that you have right now, this enterotype that you have, this is leading to your normal health status. And if you lose that, for whatever reasons, you're going to enter into this concept of dysbiosis. And dysbiosis is usually associated with something bad. And how do we reverse that? That will be some of the things that we'll be talking about later on today. And then I'm going to take a hard pivot after we talk about the gut microbiome. I'm going to be talking about different brain structures, why we eat the things that we do, and it'll be more of a psychosocial sort of look at the different brain structures and what we think happens when we change our eating habits that can impact the gut microbiome. And this is where we'll talk a little bit about the neural circuitry of dopamine as well as rewards and this concept of dynamic contrast. And then I'm going to come all the way back and then begin this conversation about not just differences in behavior, but how a change in your gut may change the pathology in your brain of things like Parkinson's, which is the most well-established, potentially autism, as well as Alzheimer's disease. And again, very controversial. Like, none of this is set in stone. It's moving and changing all the time. And this is why we call it a unicorn. We think it's real. We think that it actually, or we're not sure it's real. We're looking for it, and we're trying to get this evidence that, in fact, this is all correct. So here is this slide that I wanted to go back and revisit a little bit. This idea that Rudolf Tanzi had at Harvard University, that when he started examining the pathology sections of the brains of individuals that had Alzheimer's disease, so they were diagnosed with Alzheimer's disease, and then when he went back afterwards and he examined their brain sections, he noticed that a large number, not all of them, not 100%, but a large number or proportion of these individuals that had Alzheimer's also had resident bacteria within their brain. And it wasn't something that was added after to the slides, it was there, pre-existing. And so he came up with this hypothesis that is now gaining traction, that in fact within the brain it is possible for bacteria to live, survive, potentially also replicate and continue to live in that environment, and that over time it may result in the pathologies that lead to neurodegeneration. This is one hypothesis for how bacteria could cause disease within the brain. It has nothing to do with the gut, has nothing to do with the gut microbiome, but this is where people started getting interested in this bacterial hypothesis. There were some things that I didn't cover in the last lecture that I wanted to make sure that you were aware of and that you need to be comfortable with. So we know that neurons normally have this protein called amyloid precursor protein, and amyloid precursor protein is usually processed into things that are toxic or believed to be toxic, like beta amyloid or amyloid beta. And this amyloid beta, we know that there are different proteases that exist within your brain that could cleave it, but it's also been thought that different types of pathogenic bacteria, including the Borrelia species, could also have proteases that could assist or lead to even faster accumulation of the breakdown product of APP known as amyloid beta that you see here. In addition, these bacteria may also help to let the amyloid beta evade the immune system by creating a biofilm which is resistant to immune attack. So there are lots of ways this bacteria could actually help in the pathogenicity of the development of Alzheimer's. One of the cells that we're going to be talking a lot about over the course of today as well as in next week's class, and we're going to revisit it several different times because the microglial cells that you see up here are believed to be incredibly important. And in fact, if you're looking at health and disease or you're looking at neuroscience, a large proportion of the research kind of has to look at the role of these microglial cells. And one of the things that we think also happens is that whether it's through the amyloid beta or whether it's through a combination of interacting with bacteria and amyloid beta, these different factors like amyloid beta and or the pathogen are able to activate on the microglial cells a receptor known as the toll-like receptor 2, or the TLR2 as abbreviated here. And this will then set off an intracellular cascade in the microglial cells that will eventually lead to the activation of NF-kappa B. It will eventually lead to the production of different types of cytokines that assist or make it more inflammatory within the brain. So they release, after the activation of the TLR2 receptor, more of the pro-inflammatory cytokines. So without doing a Google search, without checking up on Siri or anywhere else, those of you who have taken like M250 or any of the other immunology courses, what types of bacteria normally stimulate the activation of the toll-like receptor 2? There's a specific class. Do you guys remember, or have you learned that? So it is bacteria. And there's either gram-negative or gram-positive bacteria. Do you guys know which one it is? So the gram-negative types tend to stimulate this pathway more. And so these Borrelia tend to fall into that type of category. So there's a lot of interest in this whole pathway. Now, I wanted you to take a look at this sort of question here. So if we believe that this is true, if we believe that bacteria are somehow, and don't ask how it gets up into the brain. Like even Rudolf Tanzi does not have a good explanation for it. Are you born with this after, or are you born with it? Is it part of your brain right from the beginning and you just happen to be unlucky and Borrelia gets in? Or does it enter later? What does that mean for the blood-brain barrier? There are so many questions. We don't have those answers. But take a look at this question, discuss it really briefly if you can, and if we were able to just eliminate this biofilm formation, which is right in the middle of this pathway, do you think that we would have an impact on Alzheimer's disease? And a lot of you have already said that, yes, we'll have actually lower incidence of Alzheimer's in these individuals. So just talk really quickly, see if you can come up with a rationale for or defend one of these different choices. Okay, so I'll give you like 2 minutes, and after about 2 minutes, I'll start taking up whatever answers you guys come up with. And you should be aware that these types of questions are the type that I'm more likely to ask you on your upcoming midterm. So, just go talk. Talk to yourselves. You guys are facing each other already. Even if you just want to say hello, like you've never met the person in front of you, just go ahead and start discussing like what you think might happen. All right, so I think most of you are coming up with a consensus. Are there any groups that are willing to share what their thought processes? So a lot of you have chosen the wrong answer. So any of you willing to share your thought processes? So everyone's like really quiet now. Is this what the midterm is going to be like? No, it won't be. Anyone want to share? Or thoughts? Yes, please, yes.
So there was a bit of a disagreement. Some of us thought it would lower, others thought there would be no change.
Right.
Logically speaking, the biofilms protect the plants of beta amyloid. So if you stop, if you inhibit the biofilm production, that should lower DE prevalence. But then also there are two other pathways
Right.
where the pathogen are producing proteases to produce that clean APP into beta amyloid, and also with the toll-like receptor pathway.
Right. So your colleague, for those of you who couldn't hear at the back, your colleague has been suggesting that perhaps there are other pathways that are involved. Biofilms are one potential pathway, but in fact, what we're going to be learning about here today is that this essential role of the microglial cells in this whole process, in addition to the beta amyloid, because there are other pathways, you're likely not going to affect the incidence of Alzheimer's disease. It won't go lower, it won't go higher, it probably won't change at all. And the role of these microglial cells is really what I'm highlighting here. Now that was a bit of a misleading sort of question for you. You won't have to worry about that type on the term test, but I wanted you to talk to the people around you and to kind of go back and revisit this particular idea. So we have a lot of evidence to suggest that, in fact, in dysbiosis, especially dysbiosis that occurs after a change in diet, that we get a shift, and this really hard shift in the normal gut microbiome. Your enterotype will change, and this enterotype that we're going to define, instead of the 3 different types of genre, let's talk about the major phyla, the major groups. The bacteroidetes and the firmicutes, these will change in their ratio. So as you have more of a weight gain that's associated with changes in your diet, you may have a change where you get a decrease in the bacteroidetes and an increase in the firmicutes. That ratio that existed before is going to be off balance, and it's going to mirror this type of change. And so this first bullet point is a really important one for you as well. In human studies, not looking at the bacteroidetes and the firmicutes, when they looked at human studies, some of the different types of phyla are just a little bit different. And at least in one study, where they looked at a difference in weight gain and a difference in the enterotype measuring these different phyla, they actually saw a decrease in the bacteroidetes, but they saw an increase in the actinobacteria. So in human studies, it's a little bit less precise. In mouse studies, we always see a change in bacteroidetes and firmicutes. And this will be a common theme that we look at in several different papers as we go along. So again, very, very interesting that we get this shift. And this shift is actually probably due to the fact that you have more energy-rich resources as you change your eating habits. And as you change your eating habits to have more carbohydrates and more fats that are available for them, that you will get a shift in these different phyla. And that's represented by this. Just out of curiosity, because there is a slide on this later, how long or how quickly do you think this change can take place? So if I decide, because I do this anyway, so I'm not a good example. So someone really healthy in this class, you work out all the time, you are really careful with like what you eat, and you really take care of your diet. If you go out and you have a burger, or a burger that you normally wouldn't have, and you have like a milkshake, and then you also decide to stop off at Tim Hortons, and you decide that you're also going to have like one of those ice caps, and then you decide that you're going to go to Dairy Queen and have like a sundae, will you change your gut microbiome? Sorry? Immediately, like right away, on the spot. So within 24 hours. And again, there's some evidence from some labs that the human gut microbiome can change in as little as 24 hours. Does it stay that way? It doesn't. It'll go back if you stop eating those foods, but it does show you that there's a lot of plasticity within this gut microbiome and these different ratios. And again, if you just change the gut microbiome, so you're not changing the eating habits, you're not changing the types of foods that they're eating, you just replace the gut microbiota from an obese animal into one that is lean, you can actually induce obesity. And that's actually fairly consistent in the literature now in 2019. Many studies have actually shown this, and this last bullet point were some of the early studies about 10 years ago that actually did that. A gut microbiome transplant is enough to induce a physiological change in this animal. And again, this is sort of the change in ideas that we've been having. This is enough to cause a physiological change. Again, I don't want you to stress over this figure. There's nothing that's going to be tested on this particular figure. This is just to go back and reiterate that we have so many different things that we now know can impact on the gut microbiome. One of this is age. So your gut microbiome has changed from the time that you were born into your first year, into like your second year, and then into your adulthood. That's changed. And as you get older, it will change again. And this is what this is showing you. If you happen to have some kind of disease process, your gut microbiome will change. If you alter your lifestyle, so you stop exercising, your gut microbiome will reflect that. And so all of these factors play a role, including the different types of drugs that you're taking. And we never used to think about that 10 years ago. And now we're very, very concerned about that. So no one used to think about, oh, you're on antidepressant medication. And these antidepressant medications may actually be altering your gut microbiome, which make things worse or better. And so we're kind of going back and revisiting all of that right now. So I'm going to segue into sort of the next study where we're looking at the particular role of diet on the gut microbiome. So in mouse models, where most of this literature has actually come from, so 2006, 2008, 2009, there were some initial papers that really caught the attention of a lot of researchers. In these mice models, so if you shift from a low fat and plant polysaccharide, so fiber-rich, not processed monosaccharides, but plant polysaccharides. So these are good diets, to one that is more refined, that has high fat, high sugar, or what we would call a westernized diet. You're able to change the bacterial content within a day. And again, this seems to be fairly consistent within the literature. And also similar studies in humans have shown that within a day, using different types of sequencing techniques, we can actually see changes in the gut microbiome in humans within a day as well. So all of this suggests that we can change this gut microbiome. Changes in the gut microbiome might be associated with different forms of disease processes. It might be associated with really severe neurodegenerative conditions, and we need to understand how these differences in the gut microbiome can play this role. And again, these changes in diet have been well characterized. Okay. So I'm going to show you a couple of figures. These are important, and I won't lie, you're going to need to know them, and you're going to need to know some of the different parts of the slides that I'm going to show you. If you did read the announcement, there was a paper that came out today, like just literally about an hour before I showed up to class. There was a paper that came out in, I think it was Nature, and that Nature paper actually revisits this same controversy. So it's kind of an interesting hypothesis about where do these bacteria come from. And you might think in 2019, and we're going to have this discussion in a moment, why is there still a controversy, right? So we need to understand that. There is no consensus. Half the labs still, in 2019, if you read that paper, and you don't need to, but if you are interested, read that paper, half the scientists believe that the gut microbiome is established after birth, and half the scientists believe that it might actually begin before birth. And there's a lot of controversy about this. So is the in utero environment completely sterile? We used to think that. That's what I was taught in undergrad myself. We now know that it may not be. So for example, a good case in point, Outside of the brain, what other organ system do you think should never be infected with bacteria? Because you've heard that having bacteria in that organ system is bad. Anything? Go back to your physiology, organ systems. The respiratory systems. Having bacteria in your lungs is a bad thing, but we now can detect a lung microbiome. Prior to 2018, nothing. But now we are able to detect bacteria growing in there. And so we have to think a little bit about this. Why is this the case? And so some of this controversy stems from the techniques that are used to detect where these bacteria are coming from. So some researchers using different types of techniques have detected bacteria in healthy neonates as well as within the amniotic fluid. So if you are able to detect bacteria in the amniotic fluid around the developing fetus or the just before birth, then it suggests that there's bacteria in there already, and that's already seeding the gut, maybe. Other individuals, when they are looking at the meconium, which is the first poop that a baby makes, so the meconium, if you see it on your term test, the meconium just refers to the first bowel movement that a baby makes after birth. And one of the things that you should be able to detect, because it's coming out of the gut, if it's been sitting in your gut, and that's the first poop coming out of the gut, and you have bacteria in your gut, you would suspect that you would be able to detect bacteria in the meconium. But so far, most scientists have not been able to detect bacteria from the meconium. It's free of viruses and very, very minimal or no bacteria. So that's where this controversy is coming from. Does it exist beforehand or not? And these are sort of the scientific proofs of this. This last point is one that if you're really interested in it, some of you were mentioning that you had relatives or individuals that have irritable bowel syndrome, or you have different sorts of IBS-related conditions. I just put that up there in case you're interested in that process, but we're not going to concentrate on some of the physiology on that last bullet point there for you. So here is from a previous slide some of the things that I mentioned to you earlier, and today's paper actually is trying to address one of the other controversies. So the first controversy is, are you born with a gut microbiome to begin with, or do you develop it after birth? And we're going to try to address this here. This current paper is actually trying to address, if you are born by C-section, does it matter? Does the gut microbiome matter, and is it different? So there's no doubt it's different, but does it matter? That's the key finding in this particular paper in 2019. So let's assume on this slide here on the left that what we're looking at is a sterile GI tract. So let's make the assumption that because of the meconium being free of different bacteria, we don't have anything growing in it, and it's sterile. There are no bacteria there. We'll make that assumption. It may be wrong. You might come back in like 2 years and say, and bring out this fantastic science paper, put it right up to my face and say, see, Dr. Ju, they have proved that, in fact, there is bacteria in the gut at the time before birth. But let's assume for the time being in 2019 that there is still a sterile environment in utero. So before the baby is born, there is no bacteria. This is now well established, and this was established as early as 2011, and this review paper in 2012 is just summarizing what we now know. And this 2019 paper is actually not adding anything new to this particular data. But if you are looking at the time of birth, and then you look 1 month or a couple of weeks later, and then you were to sample the stool, one of the things that you would see is if you were born by C-section, so you were removed from the uterus surgically rather than having a vaginal delivery, the species of bacteria that exist in your gut within the first few weeks to the first month are completely and dramatically different than if you were born vaginally. So vaginally, you would have in your gut microbiome, you would have things like lactobacillus, you would have Prevotella, you would have Schneithia. as opposed to if you were delivered through C-section, where you're in contact with like skin and the skin of like people handling you, your mother's skin, you are more likely to have Staphylococcus, Corynebacteria. So if you've studied other microbiology courses, you'll know that your skin has a lot of that. So it makes sense that your gut will be populated by bacteria that's come in contact with skin, as well as the Propionibacterium species as well. The surprising thing is, and this makes, you have to think about this, right? And this is one of the questions that's going to come up next. So you start off with very, very different bacteria. And that's, again, well established. And then after sort of the first month, everyone resets. So everyone resets to Enterobacteria. So does that make sense to you? Everyone resets back to Enterobacteria. You started off with completely different populations, but then everyone gets Enterobacteria. How many of you like that? How many of you don't like that? How many of you can raise your hand? No? Oh, okay, a couple of you. Awesome, awesome. So to me, it's really bothersome, right? So this says that at least in early development, the gut microbiome can't really establish itself well. And then it doesn't matter if you're fed breast milk or if you are fed formula, everyone eventually converts into this Enterobacteria. And again, it's really surprising to me if you're thinking about the importance of food and diet, everyone converts into this enterobacterial pathway. At about 1 to 6 months of age, though, we start to see a further division where we start to see these bifidobacteria and these bacterioids that we saw last week start to make their appearance at this time frame. And then once you start introducing solid food, everything changes again. So we've had this initial colonization. We've had this transition period leading into this bifidobacteria, so the second major change to your gut. And then once you're starting to eat solid food, this solid food will again change your gut microbiome. And this gets into what we are a little bit more familiar with, the bacteroidetes and the firmicutes. And this ratio of the bacteroidetes and this firmicutes, these phyla, are the ones that are important throughout the rest of your life. And again, depending on what happens to you, and none of the things on the right in adulthood, so we'll come and talk about that a little bit later, but none of the things that lead to these changes, you don't have to know that allergies will change to increase the level of proteobacteria, et cetera. I want you to just be familiar with the fact that into your adulthood, so when you started eating solid food up until this point in time, your gut microbiome has pretty much stayed the same until something bad happens to you. If you change your diet dramatically, et cetera, this ratio of the bacteroidetes and firmicutes will change. Now, do you have any questions before we go on, or anything that is not clear? Yes, Isis. Right, so this is a great question that one of your colleagues was asking. So what if you don't have proper nutrition and you're malnourished? Does this process still happen? It does not. So this is assuming that you have normal levels of nutrition, but in malnourishment, this actually never sets. There's another question at the front, I think. Yes? So that's a great question, and it's a question, is there any remnant when we start changing over? There's a lot of suggestion that there is, that we always have sort of a remnant of this left, and we're going to talk a little bit about that. And let me just check really quickly on the next question. So I need to know a little bit about your background, so if you can take a few minutes, talk to your neighbor, and figure out which one of these different methods you would use, because right now we've been talking about all these different changes that we've been able to identify on this slide here. So we've been talking about the shift that's going on from like the time you're born into your first month into like when you're eating solid food, and now we've been talking about this change in ratio. What do you think allows us to make these types of claims? What do we do with the different stool samples? Okay.
But I love Jeff.
All right. Thanks for yeah. Okay. All right, we'll wait till we get to about 150 on here, and then we'll sort of take up the question. All right, so boy, your class is like really interesting. So the first question, everyone except for one person got the wrong answer, one that was chosen the most. How do you think you guys did on this question? Doing better? Much better. Okay, no. Gram staining is really useful for identifying different bacterial species, but it can't actually identify the differences between bacteroidetes and firmicutes. So if you want to rethink that a little bit, where would you reshuffle like your answer? So that option that most of you chose is not a good option. So what would you choose instead, perhaps? All right. So I see actually more of you shifting over to the answers that you are seeing up here. So we'll just sort of cap it for the time being. Most of the data that I'm going to be showing you today, and you should be aware of this because you'll need to know it not only for today, but also into next week. You'll need to know it for your term test. The quick, dirty, easy way to identify different bacterial phyla is to use what is known as 16S ribosomal sequencing. However, that was the quick, dirty way, and now that next generation sequencing is faster and cheaper, more and more researchers are moving toward next generation sequencing of the gut microbiome. It's still more tedious, but it gives you a much more detailed map of individual bacterial species and genre than just looking at the phyla. So people are right now kind of moving from the 16S ribosomal sequencing, which is the correct answer, into next generation sequencing, which is also the correct answer. You need to know that for the upcoming term test because you're going to be asked how you might be able to detect these changes in the gut microbiome. Okay, so these are the changes that we saw up here. How do these changes actually get initiated? And this gets back into like pure familiar territory about the reward circuits that all of you are familiar with. So we know that different circuits that exist within your brain actually will change your behavior. In order for you to show up to class, you need to find it rewarding. And when we need to stimulate that reward pathway, it means that we probably need to stimulate one of the pathways that I'm going to be showing you, whether it's the mesolimbic or the mesocortical circuit. We need to stimulate that reward pathway for you. So food-seeking behavior is often associated with a reward or safety, survival. There's some intrinsic rewards associated with that. All of this takes place in the midbrain area of the brain, and we're going to talk about that circuit in a moment. So if we look at how other things inappropriately alter this reward pathway, we will see a change in behavior. And you need to make this link, and it's not a really good transition, but I'm hoping that you'll see why I made this transition. So what we've been talking about is an alteration in the gut microbiome. And you can alter this gut microbiome by changing the type of food that you eat or changing like your health, exercise routines, etc. And how do we actually initiate this change in behavior like eating? We would make use of this mesolimbic or mesocortical pathway, these midbrain circuits that I'm going to be showing you in a moment, because altering this does really alter how you eat, how you seek out different rewards, etc. And we know that drugs of abuse, especially if you talk to people who are very strong drug abusers who've been chronically using these different types of drugs, they will say that they're hungry for like a taste of cocaine, or it feels like it is like sex for them, or that cocaine really tastes really good, or it stimulates like a certain visceral taste response in them. So these different rewards are reinforced by the way that we also kind of talk about them. So these are the different reward pathways, and I'm sure that you've seen them several different times. And I wanted you just to be aware of these pathways. I'm not going to be testing you on these. I'm making the assumption that somewhere in your second year neuroscience class that you've learned about the VTA to the nucleus accumbens, and you've learned a little bit about the release of dopamine onto these pathways, and they are important. And these lower pathways, these mesolimbic pathways that control circuits within the amygdala that will feed back are all important, but we're not going to really concentrate on them. I wanted you to be aware of them because they're important in our discussion, but you should not expect to see a question or to be tested on this material. So we're looking at this dopaminergic system, and this dopaminergic system involves the nucleus accumbens and the VTA. And again, if we take a look, a lot of the different types of substances that seem to mediate a change in reward-seeking behavior are things that are addictive. Things like opiates, alcohol, nicotine. These are drugs that will alter this pathway within your brain. And then if we were to take a look, there would be other types of drugs as well. And again, I know you've covered this circuit. It's just a review for you. In fact, we're going to come back to this circuit later to talk about the genetic long-term changes that go on in this pathway in depression, not now, but later when we talk about depression. It's the same circuit, this dopaminergic circuit, that tells you that something is rewarding. We're going to come back and talk about it, but you don't need to worry about any of this information on this slide. It's just to orient you a little bit and also maybe to get you to think. We have certain drugs that we've isolated that can stimulate this pathway, increase the availability of dopamine. Dopamine is the key driver, and so does everything work through dopamine? And that's really the key question on that slide. So the previous one, don't worry about all of the details on there. We know drugs like cocaine, amphetamines, THC, and others, even nicotine will target different aspects of this reward circuit, the VTA to the nucleus accumbens. Again, the data on this slide, not important to know that some work more on the VTA, some work more on the nucleus accumbens. It should all be a review. And I'm just trying to make sure that all of you are aware of why I'm talking about dopamine and the different types of addictive drugs work on this pathway. Are we all clear on that? No details, just sort of background. You know, it's fairly specific background, but I need us all to be sort of on the same page. So dopamine is really important for motivation. It's important in this circuit. It's important for reinforcing different types of behavior. I've already made sort of the link on activating this pathway and linking it to behaviors that sound a lot like eating. So taking cocaine isn't eating, but a lot of addicts that take cocaine on a routine basis, they will talk about it like eating, like I'm hungry for that, or it tastes so good, etc. So this dopamine pathway is an incredibly important one, and this dopamine pathway is one that we need to understand when we alter a behavior that can result in a change in physiology or a change in brain structure. And one of the reasons why this is so important and why I'm bringing up this pathway is this diagram here. If you have studied anything in neuroanatomy, this mesolimbic pathway does now have connections to another structure which is known as the lateral hypothalamus. And this lateral hypothalamus is involved in your basic behaviors. Sleeping and eating, etc., are controlled by this lateral hypothalamus. And it feeds into that circuit that is important in reward, that drugs of abuse co-opt and kind of overactivate in inappropriate ways. The lateral hypothalamus can also do the same thing. And the lateral hypothalamus is a thing that the group of neurons that actually feed into this pathway that make you want to eat, that make you want to drink, and make you activate this dopaminergic pathway to begin with. And so when people started to look at this, and it's still controversial, and again, this course is a little bit about observing different controversial topics. Do you agree that for most individuals, cocaine is an addictive substance that will alter dopamine levels? Yes. Okay, good job. So I hope you all agree. Do you agree that drugs like nicotine can also be addictive for some individuals, also altering the dopaminergic pathway? Okay, not a trick question. Okay, awesome. So how about food? Is food an addictive substance? Yes. Okay, it could be. And some of you are thinking, well, it's obvious. But for neuroscientists who have been studying this for a long time, it's been a cause for debate. It's the same sort of thing. Is it real or is it something that's fictitious? Does it activate the same pathway? Because you're normally supposed to eat. How could food be an addictive substance? And how can food overstimulate this dopaminergic pathway? And it makes a lot of sense from your perspective, but neuroscientists still debate whether or not this circuit is seeing exactly the same response and the overstimulation of dopamine when food is involved. And you might think that's a no-brainer question, but in fact, it's actually a very important question. And this becomes important because we know that different types of food can actually be addictive in ways that we know work on this dopaminergic system. So what we're working on now is to try to identify how we can change the gut microbiome by changing our eating habits and how modulating this dopaminergic system can actually change our eating habits to change our gut microbiome. So are you with me so far? Okay, awesome, thank you. Is everyone with me so far? Okay, so here's where we're trying to get to. So if you were to take a look at the different systems and the different ways in which dopamine is normally involved, so dopamine is involved as sort of a reward prediction pathway. When something works out really well, or when you get a surprise, dopamine is there kind of releasing things. So when you taste something brand new that you've never encountered before and you tend to like it, that's dopamine kind of working in the background. That's that novelty sort of seeking prediction pathway that is going on. And food, being a reward, is normally part of that dopamine releasing pathway. So food normally does release dopamine, but not at really, really high levels. And so how do we get really high levels of dopamine beyond what we normally get when we're eating? Because that's part of the normal circuit. And there's been a lot of research into this. So let me ask you, how many of you use Apple products? How many of you have an iPhone? Okay, so most of you have iPhones. How many of you have like the iPhone 10? How many of you will get the iPhone 11? Why are you going to get the iPhone 11? It's kind of different, kind of, I don't know, it has like 3 different cameras at the back. I'm not sure. But part of the reason why you might do this is because it's different enough. If it was identical, so if the iPhone looked exactly the same and there were no changes and you didn't see any difference, you probably wouldn't get the iPhone. But now that it has sort of 3 cameras at the back, not just 2, 3 cameras, you might actually think, wow, it's different. It looks kind of new. And so this idea that in fact dynamic novelty, which is a psychological sort of construct, that this idea that something new that you haven't encountered before causes a little extra surge of dopamine is actually important because we use this to our advantage all the time. So for example, when you are thinking about like what you eat, some of you will find some foods to be more enjoyable to eat than other types of foods. So you wouldn't, for example, eat just plain old sugar, but you would eat sugar that tastes, that has exactly the same caloric intake, but actually has different textures, have different flavors kind of mixed in with it. And so you would probably eat a chocolate sundae, but not eat all the different packages of sugar. And so this is sort of how we work in terms of how we change our eating habits by releasing more dopamine when we eat, and that food does indeed become addictive, and by making that food addictive, it alters our gut microbiome. So I showed you a picture earlier of Gwyneth Paltrow last week, and I also showed you pictures of me eating my quarter pounder. So, do you think I still eat quarter pounders? No, I still eat quarter pounders. How many quarter pounders do I eat? One. Would it embarrass you, or would it embarrass me to tell you that I eat like 2 a day? Ooh, bad. Right. So I know that it's bad, but it's still something that I kind of have an association with, and I kind of like these quarter pounders. And so this idea that this dynamic novelty can be used to change our eating habits is a really important one. And I think right now it's probably a good time to go ahead and take a break, and then we'll come back and go into a little bit more detail about dynamic novelty and how it works, and then we will finish off the lecture. If you do need to talk to anyone from Notonic, they're here at the front, and please feel free to go and connect with them. Hello, Stephen. Hello, Stephanie.
like after blood transfusion, people. We'll talk about that. Very cool stuff. Hopefully it's right.
So I had two questions. First, we're actually talking amongst the table for a bit, so I decided to come up and ask. We were thinking that dopamine mediates reward and reinforces certain behaviors. So what if you were, what hasn't an experiment ever been run where they would pair dopamine supplements with eating food that is good for your gut bacteria that will like you reinforce the desire to eat good food?
Yeah, so unfortunately, dopamine, moving dopamine, would have to be like L-dopa. Dopamine can't cross the blood-brain barrier. It's charged in the bloodstream. It would have to be like L-dopa. L-dopa has hippocampus testing association. So you'll be able to, if you take enough L-dopa, there's enough dopamine associated with it, you will make associations.
So would that work as like a way to get people to eat better food? Like,
That's a good question. Maybe. I don't think there's been a formal study that it's a good hypothesis.
and secondly, babies inherit their mother's immunities like their vaccinations and immunizations in utero because they share blood. So is there any possibility that the enterotype will also be inheritable?
Some indications that mother's gut enterotype is the same, but in fact it turns out that even if you have some kids living in your house, their enterotypes are so much worse because they're eating the same food, exposed to the same environment, they're eating the same bacteria. So until a properly controlled study is done,
How would a properly controlled study look in this case?
so I think they would have to start with a mouse model and talk with some of the different mice models and then move into like human studies. But it would be unequal to do something big all the way. So it's maybe
Oh, right.
socially isolated kids.
No, you don't. You need to socially isolate them to test that.
So I don't think that would be. There might be ways around it, but that would be like the most focus for them. Do you have a quick question?
Oh yeah, sorry, well, the light bulb on our desk is flashing. I don't
that's
know
okay.
like what is it. Okay, thank you.
Stephanie. All right. Why are you still here, Marlene? Why are you still here? Why are you still here?
Just hanging out if anyone needs me. I don't have class today, so.
You can hang out with like Emilio and
Sorry?
you can hang out with Emilio and some of the other fourth years that are at the back there.
Here? I didn't know.
I didn't know either.
So I didn't pop in there.
Okay, everyone. If we're okay to go. So lots of great questions, lots of great discussion. And again, I'll try to give you my own notes and sort of summaries on what I'd like you to take away from today's lecture in a separate document that I post a little bit later on. let me try to get this back. Sorry, so I think it's... Oh. So Jeffrey, if you're around, how do I get the screen back? I think it's HDMI 3. Yeah, and then this one there. Okay. Okay, awesome. Thank you. All right. So we were talking a little bit about how using this dopamine system allows us to kind of go in and change our eating behavior. And ultimately, we want to see how changing an eating behavior, which may or may not be something that you want to do, is something that can affect the gut microbiome. Sometimes you want to change your eating behavior for health reasons, and sometimes it just happens that you start eating junk food and it becomes a process where it gets harder to stop because of this dopaminergic system that is being activated. So this is that particular scenario of like how this system is being co-opted or overrun to start you into like unhealthy types of behavior. So for those of you who are big fans of Tim Hortons, I'm not sure if you like the Tim Hortons coffee. If you do go to Tim Hortons, how many of you drink your coffee black? Do you like black coffee? How many of you at least have to have like a double-double? Can't get by on just like black because it tastes kind of bitter a little bit, and the caffeine tastes bitter because it's an alkaloid, so you need that. So at one point, not too long ago, about 3 years ago, I realized that I had gone from asking for a medium double-double and asking for an extra large quadruple. And if you've ever seen a quadruple, that's a lot of cream and that's a lot of sugar, but it sure tasted good. And so this is sort of where I'm coming in to tell you a little bit about why certain types of processes that work on the dopaminergic system can actually be really bad, and then going back and talking about this concept of dynamic novelty and how this dynamic novelty actually works in the real world. So one of the first people to work with this is Drunowski, and Drunowski actually started looking at this in the late 1980s into the early 1990s, and he wanted to know, like, why do people, or is food addictive? Does food work on this dopaminergic system? And why do different types of food become more addictive than others? And so one of the things that he started to do was he wanted to see if he could change the eating behavior of different types of animals. And so he would provide animals with a certain amount of food, and they were all given the same amount of food. And one of the things that he started to do was he wanted to see is if I wanted to get an animal to eat a certain amount of food, how can I change their behavior? And so one of the things that makes an animal eat more is by changing the amount of fat that is actually incorporated into the food. So if you go from pellets that contain like very, very low amounts of fat, like skim milk, and then you put in like more and more cream in the food chow that they're eating, they will actually eat more. So by simply changing the amount of fat that is associated with it, you can also change the eating behavior. So they will eat more of the food pellets, and they're eating more of the food pellets that contain more of the fat. And they start to choose and prefer eating things that have more fat associated with them. He also found that it was also really important to maintain the balance between the sugar as well as the fat. So he wanted to make sure it's not because it tastes any sweeter, etc., and it was simply due to the amount of fat that was in the food that the animals were given. And this idea that by having this increase in texture, because fat actually changes the texture of the food, led to this idea that he developed, which is this contrast. The fattier the food is, the different it tastes, and it's a novel type of experience for the animal. And this led him to get this idea, this dynamic novelty or dynamic contrast that allows us to eat more in addition to having the fat that's in the food to begin with. And this is, again, if you like eating, do you know what cookie this is? So the Oreo cookie, #1 best-selling cookie. Why is it the #1 best-selling cookie? Because it tastes delicious, of course. But why does it taste delicious? So how many of you take apart the Oreo? Be honest, be honest. Some of you take apart the Oreo. So what do you eat first? Do you eat the cream filling first, or do you eat the outside parts? The filling. Okay, so what does the filling taste like? It's pure sugar. Okay, right. And it's fatty sugar, right? So it tastes good. But what are the cookie wafers? What do they taste like? They taste like paper. No, they do not. They do not taste like paper. What do they taste like? Not really. So I think we should do this experiment next class. Shall we do this experiment? We'll bring in Oreos. We'll all take them apart. They taste a little bit bitter because it's not sweetened chocolate, and it's like cocoa. So you're getting this contrast between something really sweet and then something slightly bitter on the outside, and it tastes better when you eat it together. And we'll do that test. We'll see if that's true in the next class. And this is that idea of this dynamic contrast. So let me show you an example of an experiment, and you should start to take some notes down here because this is a really interesting experiment on how addictive this can be. So what we're looking at now is dynamic novelty. Dynamic novelty can be used to change eating behavior, but I haven't really shown you that it can be addictive. And so let me show you why it can be addictive. So one of the experiments also done by Drunowski's group was a little bit different. So he started to look at these Sprague Dawley rats, and these are a species of inbred rats. These are white lab rats. And these white lab rats can be fed on a high-fat diet. And if they're put on a high-fat diet over a 2-week period of time, you can actually separate out these two rats. So again, we've talked about including fat will generally make an animal eat more. And that was the previous experiment. And that increasing the amount of fat changes the texture of the food. It tastes and feels a little bit different. And so that dynamic novelty increases the amount of dopamine in some of these rats. And so that makes them eat a little bit more, and that increases their feeding behavior. But here's the really interesting thing. If you take a group of rats and you look at 24 rats, 12 of them will react just the way that I've talked to you about before. So they like the increased fat, they see the dynamic novelty, they increase eating, they increase dopamine, and then there's this other group of rats over this 12-week period of time that actually still eat the fat-laden food pellets, but they don't overeat. They just eat enough. And so we separate out over this 2-week period of time one group that becomes diet-induced obese, so they eat more. They like the fat, they get the dynamic novelty, they release more dopamine, they eat more and more, and they become diet-induced obese. And then there's this other group of rats over 2 weeks, we can separate them out, that actually don't gain any weight. And they are resistant to the obesity, and they become obesity resistant. Pretty cool. It just shows you that there are lots of different factors that are at work even in genetically inbred experimental animals. But here's why this becomes addictive. And here's why another layer of this dynamic novelty makes it really, really scary if you think about it. So this group of rats has resisted eating more up until this point in time. So you have these two groups, they self-select. One group becomes obese because they eat more. They've already fallen into the trap of dynamic novelty, dynamic contrast. They eat more dopamine release. This other group has ignored it. They've stayed healthy as long as they possibly can. They've resisted temptation. They're not eating more. And now you switch things up on them. So now you go back to that concept of dynamic novelty. After this 2-week period of time, is it only the fat that induces dynamic novelty? In his first experiment, Drunowski actually kept the levels of sugar the same across the board. But here, what he did was he actually switched things up. So now, he gave them the same level of fat that led to the diet-induced obesity, but now he also put high sugar into the food pellets. So now he has increased the level of sugar, And now he's giving these two different groups that have separated out over this 2-week period of time, that have been eating the same food, high fat, over the past 2 weeks, and now they're getting high fat plus high sugar. And the group that had resisted up until this point in time now is going to start eating more. And so they are now also susceptible to this dynamic contrast or this dynamic novelty. So if one thing doesn't get you, another aspect of this will. So this diet change, where you now introduce another variable that makes it more appealing and you release more dopamine because of this change in something that you haven't encountered before, actually led to this change in diet and this subsequent change in their metabolism. So this is sort of this concept of this dynamic novelty at work to change this feeding behavior. Do you have any questions on this so far or anything that I can clear up for you? Yes, Alyssa. Yes. So reversing it, increase sugar first and then increase fat. As far as I know, not in the Drunowski lab, but other groups have done that and they see similar effects. So it's not the order in which you do it. It's this concept that you're introducing a new variable that causes a release of more dopamine that increases the feeding behavior. It doesn't seem to be the case. So good question. Is there another question? If you decrease the amount of fat but keep the sugar levels the same, will you?
I guess like it doesn't make sense intuitively, but say if you start with a high fat diet and start to reduce and increase fiber, because that doesn't necessarily, because fiber isn't necessarily related to dopamine, but it is a novel.
So if you introduce a different novel variable like high fiber, can you also change their feeding behavior? That's an interesting question. To be honest, I'm trying to rack my brain if there is that type of study. I'm not aware of one, but I can go back and check. So if I find one, I'll post it on the discussions page. Yes?
So increasing the fat already created a dynamic novelty, but increasing sugar and fat at the same time.
So one group was able to resist that change. They weren't overeating, they weren't modifying their behavior, and they've been resistant to it, but changing yet another variable was enough to induce a change in feeding behavior.
So the increase in novelty was...
Right, the second change in novelty was enough to exacerbate it. Yes?
Did they say why that one group was resistant?
No, so one of the things that we're going to encounter, and again, I'll just put this out there broadly. If you've taken like other psychology courses or other neuroscience courses, we're not able to explain everything the way that we'd like to. So these are all genetically identical rats. It's like having twins, right? So they're all genetically identical. They're all like inbred strains of rats, very common, Sprague dollies. But one of the things that we see within mice and rats is this alteration in behavior. So we'll see this later when we talk about depression. There are groups of mice all exposed to the same sorts of stresses, and one group is completely resistant to that stress, and the other group is susceptible to the stress. We don't understand it. It can't be at the genetic level because genetically they're all identical. Is it epigenetic? Again, there's some evidence that it might be epigenetic, but there are other variables that we just don't understand right now. So it's a good question, but I don't have a good answer for you. Yes? Are the rats isolated from each other? In most of these experiments, yes. Are there any other questions? Yes, Sunny.
They specify what type of fat it is. For example, like there are different like good fats versus fat fats, or was it just
No, they weren't specifying. Back in the 1990s, they weren't really as interested in the types of fats, whether it was like a certain type of cholesterol or others. It's a good question, and we'll come back to that later on. Okay, so just for the sake of time, we'll go on to the next slide. And again, just another point in a slightly different way to show you that this is working on something that's very much the dopamine system. So we've looked at two different experiments involving dynamic contrast and dynamic novelty. They are working to alter the dopaminergic system. They are altering a behavior by increasing feeding. And does it work in the same way that it would work for a drug? Because that's what we started with. If this is working on the VTA and the nucleus accumbens pathway, we should be able to test it like the VTA and nucleus accumbens pathway. And one of the things that we often do in psychology and in behavioral neuroscience is to use a progressive rewards model. So if something is rewarding enough, then you will keep at it, and you'll keep at it even as the level of difficulty kind of goes up. So it's a lot like you would do in a class. So would you come to class if participation counted for like 0.5% of your final grade? My guess is most of you would, like if that was important. And would you keep doing that for every single class? Again, a lot of you would keep doing that. And it becomes harder and harder, and there's less and less reward associated with it. And this idea of progressive rewards is a way for us to test if this dopaminergic pathway is being activated, how rewarding is it before the animal gives up? And it's related to the amount of dopamine that gets released. So a typical way to do this, for example, is an animal may be in a Skinner box. They will have to press a lever. You're directly stimulating the dopaminergic pathway. And by pressing a lever, they will get some kind of reward. And if you press the lever once, you might get like one sort of stimulation of the VTA nucleus accumbens pathway. And then they will have to press it 3 times to get a stimulation, etc. And so after a certain amount of time, to get the same level of stimulation on the VTA to release the same amount of dopamine, they may have to press the bar like 25 times before they get the same sort of stimulation they got originally. So the first time, 1 press, 1 stimulation. To get the next stimulation, you'd have to press 3 times, and then you get exactly the same amount of stimulation in your brain. And then to do like the third reward, you might have to press like 10 times. It gets harder and harder. It's progressive. And so how far in this food pathway, if food is indeed a reward, how far will animals that have been accustomed to getting a certain type of food go to do the same type of progressive reward model? And it turns out for an animal similar to the ones that Dronowski's group worked with, if you have food that contains high fat and high sugar, they will keep pressing the bar in order to get that food reward, and it's at a level that is very similar to getting an injection of cocaine. So you are activating the dopaminergic pathway, and you are using sort of this progressive rewards model, and it is involving a totally different type of stimulus. It's not a drug, it is sort of the food pathway that is being activated. So it's fascinating that food can actually change this reward pathway. So I'm gonna ask you to try pressing the button way at the back. Let's see if this works. I've always wanted to try it. Let's see if it works. Does the button go on? Nope. Does the light switch at all for you? No? Where are you pressing on the thing? No, not on the knob, so right in front of it. You're at H08, right? Yeah, I just activated for you, so go ahead.
Do we know if the rats are hungry or if they've already ate or if they're full?
Right, so that's a great question. I'm going to turn off your mic if that's okay. So one of the things that they do is to make sure that the rats are not hungry, so they're not sort of driving the system, and usually they are satiated, so they've had enough. Okay. All right, so let's kind of keep that in the back of our minds for the time being. Let's go. Let me just get that off. So let's talk about what we want to get to. So by changing feeding behavior, we can change the gut microbiome. The gut microbiome normally is stable, and we can change the gut microbiome in a number of different ways, but one of them is by changing eating, and that's well established now. And I took you through a survey of different papers earlier on. And this is ultimately where we want to go. It's not about a behavior. It's about a neuropathological condition like Parkinson's disease. And Parkinson's disease is not something that you would normally associate with like your gut. And your gut is probably the last place that you would look for the origin of something that affects your brain. And for a very, very long period of time, like well into like the span of 100, 120 years in neuroscience, we would never have thought to look within the gut. And perhaps that is to our detriment. Let me try to get that mic off because I think we're getting some feedback. Ooh, that makes it worse. Okay. All right. So this is the paper that we're going to get to, and I'm going to talk about this figure like really quickly. Ultimately, in this 10% assignment that you're going to do, you're going to create something that looks a lot like this. It could be 2 panels, it could be 3 panels, like the one that you see here, or it could be 4 panels. But these visual graphics or visual summaries or visual abstracts, depending on the journal that you're looking at, actually give you a very, very quick, easy way to understand the major points in the paper. They're not going to tell you about specific experiments or anything else, but you can probably gather most of the information about what this paper is covering from the title, as well as this sort of visual summary that all of you are eventually going to do for your own paper. And so what we're looking at here is that if we look within the gut, the typical microbiota over here on the left, one of the things that your gut microbiota does by digesting different things that get into the gut, different foods that get into your gut, they will produce these short-chain fatty acids. And there are 3 of these short-chain fatty acids that are important that may go in and activate in your brain these different microglial cells. And I talked about microglia at the very beginning, and that this in turn may cause motor dysfunction, which many of you know to be important in Parkinson's disease. So that's a hypothesis. And so in this particular paper, if you take out the gut microbiota, so you have germ-free intestines or a germ-free gut, you no longer have the generation of those short-chain fatty acids, which means you don't activate the microglial cells. You can prevent Parkinson's disease, as simple as that. On top of that, if you were to take the gut microbiome of someone who has Parkinson's disease and transplanted that into a mouse that does not have Parkinson's, you can actually induce Parkinson's-like phenotypes. And this is sort of what makes this paper so amazing. And I'm hoping that this visual summary encapsulated all of the ideas in that paper. So is that clear for everyone? This is ultimately what you want to do with the paper that you decide to choose. So let me take you through a couple of things about this particular model. I may not get through all the slides today, so we'll stop at some point, and anything I haven't covered, I'll get into in the next lecture. So one of the things that we're currently looking for, and this will become a big part of the paper, in modern neuroscience, if you're looking at neurodegenerative diseases, we are always looking for a protein. And we're always looking for a particular type of protein. And it doesn't matter what neurodegenerative condition you're looking at. All neurodegenerative conditions all boil down to some kind of protein, something goes wrong. And in Parkinson's disease, one of the things that we're looking at more often than not is a protein known as alpha-synuclein. And something goes wrong with alpha-synuclein. We've already looked at one protein that goes wrong in Alzheimer's. It's beta amyloid, or it might be tau. But there's always a protein that something goes wrong. In different types of neurodegenerative diseases that involve prions, something goes wrong with the prions. They start to accumulate or misfold, etc. And so all of the modern theories of neuroscience and neurodegeneration go back to identifying this misfolded protein that is a hallmark feature of all of these. So it doesn't matter what disease you look at. Huntington's, we have the accumulation of these nucleotide expansions that lead to different proteins accumulating. Parkinson's, it might be alpha-synuclein, for example. We all have this characteristic protein. And as shown at the very bottom of this particular slide, in Parkinson's, it's thought to be this aggregation of following up on that involves different parts of the cerebellum and midbrain. We have alpha-synuclein accumulation, and even Lewy body diseases also have accumulations of alpha-synuclein as well. So Parkinson's disease is actually a really, really serious disorder. It affects a lot of individuals in Canada and in the United States. And some of this involves changes in how the muscles move and the signals that go to these different muscles to control movement. So oftentimes you will see what is known as resting tremors. So at rest, it's not that their muscles are rigid, it's just that they can't control the fine movements of their muscles. So you'll see someone with this resting tremor like on the TTC. Or they might have trouble moving, so they'll have, like, they can't control their legs and their lower muscles, so they'll have this muscle rigidity and they'll shuffle their feet an awful lot. They don't have this nice free movement of their legs. And they will have slowness in movement. When they're reaching for something, not only are they going to shake, but they're also going to be moving much, much more slowly. So this affects the brain, and it's very, very important to keep all of this in mind. This hallmark feature is alpha-synuclein, and we now know alpha-synuclein aggregation, the clumping of alpha-synuclein is a characteristic feature of Parkinson's disease, Lewy body disorders, multiple system atrophy, a whole bunch of things, but Parkinson's is one of the most often aggregated types of neurodegenerative disorders. So if we take a look, we'll often see a progression. So it goes from alpha-synuclein monomers on the left. We have alpha-synuclein. You all have alpha-synuclein. It's an important part of your cytoplasmic proteins. They exist as monomers. They don't clump. And then they'll start to misfold for whatever reasons, and they will start to form these long beta-pleated sheets that form these aggregates, which are called oligomers. These oligomers, again, tend to aggregate with each other. They will look a lot like amyloid fibrils. These will be inside the cell. And in fact, if you look at the histology section over here on the bottom right, you'll actually be able to see these really dark rings inside this cell. This is one cell. And inside this cell, you will see these dark inclusions, which would be these Lewy bodies. So this is like the end stage for this particular dopaminergic neuron. And they tend to be found within these dopaminergic neurons. And these dopaminergic neurons tend to be particularly vulnerable to alpha-synuclein aggregation. So you guys are all good neuroscience students, right? No answer. No answer. Sure. Oh, nice. Okay, awesome. So how long has Parkinson's disease been around for? You guys are good neuroscience students. Tell me, how long has Parkinson's disease been around for? 4 to 5 years. 25 years. 200 years. 60 years. So you don't believe that Parkinson's disease has been around since we started walking? No? Wow, fascinating. Okay, like that, you're the first class that's ever said that. Okay. So here's this really interesting thing. Oh, and by the way, let me just mention, some of you were asking about this. Most of you know that we lose these dopaminergic neurons in Parkinson's. We lose them within the substantia nigra in particular. We don't really have an effective therapy. It affects many millions of Canadians as well as Americans. And one of the things that makes this particularly bad is that we can't do dopamine replacement therapy. Dopamine, if you inject it peripherally, cannot cross the blood-brain barrier. It becomes a charged molecule, and so it's excluded from the blood-brain barrier. But L-DOPA can cross, so we use that to replace dopamine. And this other drug, which is used in the early stages of Parkinson's disease, bromocriptine, is an analog or an activator of dopaminergic receptors. So it kind of acts like dopamine, but it is not true dopamine. But as you continually lose more and more dopaminergic neurons, things look more and more grim. And one of the reasons why I asked you about Parkinson's disease is we don't really have a good explanation for it. So this is 2019. You are children of the genomic era. You were born in the year that right before the draft of the human genome came out. You were kind of born in the 1999, 2000 era. You are children of the genomic era. For you, this should be like insulted. You should be insulted by the fact that you don't know the cause of Parkinson's. Are you insulted? Yes, okay, awesome. You should be insulted. You should be like hopping mad that we don't know what Parkinson's disease is caused by. We only know about 10% of what causes Parkinson's. The vast majority, you may find out 10 years from now, but we don't know right now what causes Parkinson's. 90% of the cases, we have no idea when they walk into the neurologist what caused Parkinson's. We don't have a genetic cause. We have no idea of the mechanism. And this has bothered scientists for a very, very long period of time. They've been asking themselves the same question I asked you. How long has Parkinson's disease been around for? We don't know. So has it been around since the time we have been human? We don't know. But we do know that James Parkinson is the first physician to actually describe Parkinson's and record Parkinson's, and that was in the late 1800s. So we know that it's been recorded for at least 100 to 110 years. Did it exist before that? Again, we don't know. Just because it wasn't recorded doesn't mean it didn't exist. But it's been really fascinating because we can't actually determine the genetic cause. And up until late summer of last year, people were kind of going back and forth on what actually causes this. And we're now pretty sure that at least the genetic forms of Parkinson's, because you are children of the genomic era, for some individuals who have Parkinson's disease before the age of 40, that we can trace this back to mutations in either alpha-synuclein or in Parkin. And that is now like rock solid as of last summer. And we know that this is true. But for the 90% of cases, we have like no idea. We can only identify and characterize different things that are going on. So we know that Parkinson's normally affects dopaminergic neurons, but it doesn't only affect dopaminergic neurons. Any neurons that have pigments associated with them, like the locus ceruleus, it's a pigmented neuron. Parkinson's disease also affects them. We see alpha-synuclein in pigmented neurons there. Oftentimes, we see it in dopaminergic neurons in the substantia nigra, and we know that it's called the substantia nigra because in addition to dopamine, these neurons also contain the pigment known as melanin. For the 90% where we don't know the cause, the typical onset is in midlife 50s to the 60s, and we have no idea what caused that Parkinson's disease to occur. So very, very unsatisfying, very, very angry why we don't understand this disorder. We know like where it occurs, we know the neurons that are affected, we even know some of the pathways through the genetics, but for like 90% of patients, like where you got it, I don't know. Will anyone else in your family get it? I can't tell you, we don't know. You happen to have something that looks like Parkinson's, but we can't tell you if anyone else in your family will develop it. Again, very, very unsatisfying. This again is very typical for Parkinson's. On the left, this is the band of substantia nigra neurons, and normally we have a lot of melanin within them because we have a lot of dopaminergic neurons in the substantia nigra, but with Parkinson's, we lose the dopaminergic neurons, and we lose the melanin that's normally found in these dopaminergic neurons. These neurons are important in controlling fine voluntary movements, which is why they can't walk well. They are really slow at grasping things and really slow with their movements. So, you know, for Parkinson's, you never get better. It's progressively worse and worse. So once you get that diagnosis by a neurologist, and it's most likely Parkinson's disease, you'll never have a good day again. It will just continually get worse for you, and there is no known cure. And so we go back to all of these different ideas. We do know a couple of things, though. And this slide, don't think of it as a throwaway slide. So circle this one, make some notes on this one. Make sure that you identify things on this particular slide. More and more in the early 2000s, it was discovered that changes in blood flow to groups of neurons are actually one of the predisposing factors that lead to the loss of neurons. So vascular forms of neurodegeneration are very common. So we see vascular dementia, we see vascular Parkinson's disease, we see changes in blood flow preceding neurodegeneration in many cases. So hardening of the cerebral arteries, cerebral atherosclerosis, is a very good indicator for certain forms of infection and it lasted a fair amount of time and it was untreated, if you had viral encephalitis and it was left untreated for a while, then you are also at risk of developing Parkinson's disease. I'm going to come back and talk about the link between viruses and neurodegeneration and depression later on, but it's fascinating because we don't know the cause, that we need to find new roots of the disease process. We know that different types of antipsychotic drugs, if you are on antipsychotic medication for other reasons, so you've had a mental health related reason to be on antipsychotic drugs, if you overdose on antipsychotic drugs or take too much of them for too long, you can actually induce Parkinson's-like symptoms as well, leading to the loss of dopaminergic neurons, etc. And these are some of the different types of antipsychotic drugs like reserpine, etc., that have been shown to cause Parkinson's-like symptoms. And again, I've went through all of these classic symptoms before: slowness of movement, rigidity, resting tremor. They can't stand really well. If you can push someone with Parkinson's, they will have postural imbalance. And of course, the shuffling of the feet, a characteristic feature as well. Since we don't have a good mechanism, and 90% of Parkinson's is undiagnosable, we don't know what caused it. We are looking for new ideas and new theories. So is it viral in origin? Is it something completely different? Is it bacterial infections? So people have been looking for other alternate explanations. And one of the most interesting hypotheses, if you believe all these papers that are coming out related to the gut-brain axis, then there might be a link to neurodegeneration. Again, this is all circumstantial and correlational data, so they're kind of linking the two processes, but I'm going to try to show you some experimental data in the next class that kind of makes it more of a strong link. So if you take a look overall at the data, if you have some kind of neurodegenerative disease later on in your life, before you had that neurodegenerative disease, you most likely had something wrong with your gastrointestinal tract. And there is that sort of link between neurological diseases as well as something going on within your gut. And the changes in your gut motility and the changes that might result in neurodegeneration in the CNS has been correlational, but there's very, very strong correlations with what's going on there. There is more and more evidence to suggest that there are pathways that exist between the gut and the brain, and then also back, the brain to the gut, and that these pathways might be involved in everything from anxiety, depression, changes in your pain perception, as well as ASD, which I'll cover in the next lecture as well. And in fact, if you look within the gut, 90% of your serotonin actually begins in your gut, and so it's the source of a lot of your neurotransmitters that we see. So we see neuropeptides in your gut being produced, we see different neurotransmitters, etc. We don't know what all these are doing within the gut. It's still largely unknown. Okay, how are we doing for time? So I'm probably just going to go through about 2 more slides, if that's okay, because some of these are going to be important. I'll ask you, instead of putting up a quick poll, how many of you would like to do another quiz this week and just get it out of the way and then potentially have them on the upcoming term test? Okay, so again, you can do this all the way up to the term test. There's no reason to panic that you need to do it right away. You can take your time to do it. It'll be fairly easy. And on this particular slide, there's one word that is missing from your slide, and this is important. So this word here, axenic, what does that word mean? Without looking it up. Any ideas? No looking it up. Any guesses? It's not a misspelling of axonic. Thank you. Axenic. Any ideas? Yes. Sorry, a link?
Does it somehow relate to the gut-brain axis and genetics?
Does it relate to the gut-brain axis and genetics? It does in a very big sense of the word, but what does it mean particularly with respect to the gut? It's actually one of the words that you see up there. It's either going to be germ-free or specific pathogen-free. Which one is it? Those of you who took Classic 201, remember that bird course that you all thought was a bird course and you all took it, Latin, all that stuff? Axenic. Which course? We'll talk afterwards. So this means that it's completely germ-free. This is a germ-free animal. Another way to say germ-free animal is an axenic mouse or an axenic model system. And this is going to be important. This is going to be a big part of the paper that is coming up. So we have two different ways that we can examine the gut microbiome. We can have mice that are completely devoid of any gut microbiota. They have nothing, no viruses, no bacteria, nothing existing within their gut. And we can also get rid of specific types of bacteria by treating them with antibiotics. And we call this type of mouse a specific pathogen-free mouse. This is going to be important both for your upcoming quiz online. It's going to be important for your upcoming term test because you're going to have to tell me about a model system and how you are going to look at a model system. But we can treat animals with antibiotics and wipe out certain types of bacteria, but we still have bacteria left, and that's specific pathogen-free. These mice are not axenic, they have bacteria, and the axenic mice are the ones that have no bacteria whatsoever. They are born that way, they are raised that way, and they will never encounter bacteria in their lifetime. Okay, so we're going to talk about these in the next class. And we're going to talk a little bit more about the evidence for this gut-brain axis. I will take you through that Parkinson's paper. We'll have a vote whether or not you think that that paper is a good paper. We'll discuss it in some more depth. Is there anything that I've gone over that is not clear for you before we all kind of wrap up? No. All right, so next week, if I remember correctly, we are going to have like an Oreo eating party. We will actually see if the centers are sweet, and we'll see if the wafers are in fact bitter. Have a good week. I'll see you all next Wednesday. Do you think this worked today?
Yeah, so we looked at this YouTube link online and it was like, here's audio guitar.
Was anyone watching?
There was like 12 people. It's like a decent record.